Faithfully conveying value by interpreting great books from around the world. Uh, Hi there, this is Sally from Sally's Book Talks. Today, I'll be diving into the book The Immortal Life of Henrietta Lacks. So, who exactly is HeLa? Her real name was Henrietta Lacks, but scientists refer to her as HeLa. Back in 1951, she passed away from cervical cancer. Doctors took her tumor tissue and grew it into immortal cells, which they named HeLa cells. Honestly, a lot of us benefiting from modern medicine owe it to HeLa cells. But it played a key role in creating the polio vaccine and helped scientists with breakthroughs like gene cloning and IVF. It also aided in uncovering secrets like the cancer virus and atomic bomb radiation effects. HeLa cells rank among the top medical breakthroughs in centuries. Yet, while HeLa cells became famous, Henrietta herself was forgotten for decades. Her name was missing from research papers, and her family never saw any benefits. In fact, they struggled with poverty and couldn't even get health care. For years, they had no idea that her cells were still alive after her death. It wasn't until this book was published that they found out. That's how Henrietta's story and the truth behind HeLa cells finally came to light. The book's author, Rebecca Skloot, is a well-known science writer in the US. When she was 16, in a biology class, she learned about these immortal cells for the first time and got curious about who HeLa was. But when Skloot dug around, she only found scraps of info, mostly unclear and wrong. So she decided to tell the full story of both the cell and Henrietta. So she spent more than a decade investigating, interviewing people, and digging through documents. In 2010, the book came out, and right away, outlets like the New York Times and the Washington Post hailed it as a top book of the year. It got translated into over 20 languages and turned into a popular movie by HBO. In the book, Skloot doesn't just tell Henrietta's story. She also covers the big medical advances from HeLa cells and the ethical questions that came with them. When someone dies, who owns their leftover body parts? And should their family get a cut of any profits? Behind scientific research, what recognition and respect should ordinary people who quietly donate their cells, tissues, or organs receive? Next, I'll guide you through the story of the immortal HeLa cells in two parts. First, what exactly are HeLa cells, and how significant is their contribution to medicine? Second, what controversies surround HeLa cells? All right, first let's get to know HeLa. Who was she? Why were her cells able to become immortal? Just how enormous has HeLa cells' contribution to modern medicine been? The English spelling HeLa is a homophone of the first two letters of Henrietta Lacks' first and last name. She was a Black woman born in the American South to ancestors who were poor tobacco farmers. She married her cousin and had her first child at 14. Later, the family moved to Baltimore. In her most widely circulated photo, Henrietta sports a neat short haircut, stands with hands on hips, smiling broadly, appearing thoroughly content with her life. But at that time, she had no idea cancer cells were already growing inside her. These cells would rob her 5 children of their mother while permanently transforming medicine's future. So at 30, Henrietta felt something was wrong and went to Johns Hopkins Hospital. A biopsy confirmed cervical cancer requiring immediate treatment. Back then, cervical cancer treatment involved placing radium-filled tubes into the vagina to irradiate the affected area. Yes, that radium, the radioactive element discovered by Marie Curie. Medicine then knew radium killed cancer cells, but didn't realize it destroyed healthy cells too. After nearly 6 months of agonizing treatment, Henrietta's cancer still spread. In October 1951, she died at just 31. But Henrietta and her family never knew that during her hospital surgery, samples of her cervical cancer tissue were taken during surgery. These cells were later cultured in a lab, becoming the immortal HeLa cells. At the time, Johns Hopkins Hospital was researching causes of aggressive cervical cancer and needed lab-grown cells. George Gay, heading Hopkins Tissue Culture Group, had spent 30 years trying to grow malignant cells outside the body. Most died immediately. Survivors were too weak for experiments. Behind scientific research, what recognition and respect should ordinary people who quietly donate their cells, tissues, or organs receive? Next, I'll guide you through the story of the immortal HeLa cells in two parts. First, what exactly are HeLa cells and how significant is their contribution to medicine? Second, what controversies surround HeLa cells? All right, first let's get to know HeLa. Who was she? Why were her cells able to become immortal? Just how enormous has HeLa cells' contribution to modern medicine been? The English spelling HeLa is a homophone of the first two letters of Henrietta Lacks' first and last name. She was a Black woman born in the American South to ancestors who were poor tobacco farmers. She married her cousin and had her first child at 14. Later, the family moved to Baltimore. In her most widely circulated photo, Henrietta sports a neat short haircut, stands with hands on hips, smiling broadly, appearing thoroughly content with her life. But at that time, she had no idea cancer cells were already growing inside her. These cells would rob her 5 children of their mother while permanently transforming medicine's future. At 30, Henrietta felt something was wrong and went to Johns Hopkins Hospital. A biopsy confirmed cervical cancer requiring immediate treatment. Back then, cervical cancer treatment involved placing radium-filled tubes into the vagina to irradiate the affected area. Yes, that radium, the radioactive element discovered by Marie Curie. Medicine then knew radium killed cancer cells, but didn't realize it destroyed healthy cells too. After nearly 6 months of agonizing treatment, Henrietta's cancer still spread. In October 1951, she died at just 31. But Henrietta and her family never knew that during her hospital surgery, samples of her cervical cancer tissue were taken during surgery. These cells were later cultured in a lab, becoming the immortal HeLa cells. At the time, Johns Hopkins Hospital was researching causes of aggressive cervical cancer and needed lab-grown cells. George Gay, heading Hopkins Tissue Culture Group, had spent 30 years trying to grow malignant cells outside the body. Most died immediately. Survivors were too weak for experiments. As we said at the beginning, HeLa cells were the first immortal human cells successfully cultured outside the body. Ames lab found that these cells were surprisingly easy to grow. With just nutrients and warmth, they'd keep dividing and multiplying. Gay mixed chicken plasma, boiled calimbreo extract, and human umbilical cord blood to make a nutrient broth for cell culture. HeLa cells thrived in this witch's brew-like liquid, absorbing nutrients and growing nonstop. Jay also invented a suspension culture method, using a blender to stir the cell medium so HeLa cells could float freely. With space all around, they could expand however they wanted. Day didn't monopolize this breakthrough or patent HeLa cells for profit. He generously shared the cells with other scientists and even created a way to mail live cells. He'd put cell culture droplets in test tubes, send them on planes, and flight attendants kept the tubes in their pockets to maintain body heat. That's how the cells got shipped to labs across the Americas, Europe, and Asia. Scientists who received them cultured and distributed HeLa cells using Gay's methods, even packing them in bags hauled by mules to remote labs in Chile. Scientists divided HeLa cells into batches, exposing them to viruses, radiation, infections, and bombarding them with different drugs. They hoped to find a magic bullet that could kill cancer cells without hurting healthy ones. If cells died during experiments, no big deal. Just grab more and restart, since HeLa cells grew endlessly. Today, HeLa cells are a mainstay in labs, just like lab nice. It's worth noting that other cell lines have infinite growth potential, but HeLa was the first, and most widely used, making it the go-to material. It's hard to count how many HeLa cells exist today because the numbers are huge. One scientist estimated that if all HeLa cells ever grown were piled up, they'd weigh 50 million tons, about 100 Empire State Buildings. If all HeLa cells were lined up end to end, they'd circle the Earth nearly 3 times. Henrietta herself was quite petite, standing only about 5 feet tall. HeLa cell's second major contribution was helping scientists develop the polio vaccine, which played a vital role in preventing polio. Timing-wise, it was incredibly fortuitous. HeLa cells, first cultured in 1951, proved immensely useful the very next year. In late 1951, the world faced its worst polio outbreak ever. Schools shut down, and parents were terrified. In early 1952, scientists at the University of Pittsburgh announced they'd created the world's first polio vaccine, but they couldn't give it to children yet because it needed large-scale testing on lab-grown cells to ensure safety and effectiveness. Previously, scientists used monkeys for experiments, euthanizing them after harvesting their cells, but they soon realized this approach wasn't sustainable. Back then, it wasn't about animal welfare. It was that these experiments needed millions of trials to be reliable. Using only monkeys would have cost millions just to buy them. Far too expensive. That's when HeLa cells emerged, easy and cheap to grow. They provided massive amounts of cells for polio vaccine antibody testing. In 1954, the first commercially available polio vaccine debuted, with HeLa cells playing an indispensable role. HeLa cells' third major contribution was in human genetics. In 1953, a geneticist from Texas accidentally poured the wrong liquid into HeLa cells during an experiment. Serendipitously, this caused the chromosomes inside the cells to swell up. Thanks to this mistake, scientists clearly saw human chromosomes for the first time. After further research, they concluded humans actually have 46 chromosomes, not 48 as previously thought. This corrected the long-held belief of 48 chromosomes and established the true number. So when issues arise, like extra or missing chromosomes, scientists can now identify them. This quickly helped pinpoint diseases caused by chromosomal abnormalities, like Down syndrome, which involves an extra chromosome 21. Since the 1950s, HeLa cells have been integral to major medical breakthroughs like chemotherapy, cloning, gene mapping, IVF, and viral infections. Scientists used HeLa cells to develop treatments for herpes, leukemia, flu, and Parkinson's. They even went to space on rockets to study how human cells react without gravity. If we ever conquer cancer, HeLa cells will deserve immense gratitude. Now that we understand HeLa cells' origin and medical significance, but for decades, controversies around HeLa cells have made headlines globally. The second part of my talk will focus on these controversies. These debates center on 4 main issues. First, patient privacy rights. Henrietta's real name and medical records were later made public. Was this an invasion of privacy? Second, informed consent. Shockingly, Henrietta and her family never knew doctors took tissue samples, let alone cultivated them for research. So, should individuals providing cells for medical research be informed and consulted? Third, tissue ownership rights. Are HeLa cells considered Henrietta's personal property? Fourth, commercial profits. HeLa cells have been massively monetized, generating substantial revenue. Should Henrietta's family receive financial compensation? Decades later, some of these medical ethics questions still lack clear answers. Let's start with the first controversy. Were Henrietta's privacy rights violated when her name and records were exposed? The answer, by today's standards, yes. Patient names and records must be protected. First, regarding the name disclosure, ironically, without that name exposure, Henrietta's family might never have learned the truth. From 1951 onward, HeLa cells were treated like public property in labs worldwide for decades. Yet the medical community had no idea who HeLa actually was. The name HeLa was just a label until 1970, when George Gay, who first extracted the cells, passed away. In an article commemorating him, several colleagues first revealed HeLa's real name as Henrietta Lacks and published her famous photo with hands on hips. Soon after HeLa's true identity spread far and wide, it was because of this that Henrietta's family learned about HeLa cells. Henrietta's husband and kids were initially shocked and furious. Their mom had been gone over 20 years, and they had no idea part of her was still alive. The fallout from revealing HeLa's real name was that Henrietta's family lost their privacy. Before this, nobody paid attention to her family. After the name was out, they got media attention and drew in people with bad intentions. Some called urging them to make a big scene using their Black identity. Some suspected white folks stole the Black woman's cells. Others wanted to study Henrietta's descendants, showing up to draw blood for tests. And many more constantly bugged them with questions, causing the family a lot of pain. If it were today, a cell donor's real name wouldn't be so easily disclosed. Now, regulations strictly control the link between tissue samples and donor identities. So, naming cells with the donor's initials, like HeLa cells, wouldn't happen anymore. Nowadays, numbers and codes are used to protect privacy. The second exposure came in 1985, when a journalist published a book called The Cell Conspiracy, which had a full 10 pages, quoting loads from medical records, including detailed accounts of Henrietta's autopsy after death. Seeing this, Henrietta's daughter, Deborah, broke down. She kept crying, and whenever she closed her eyes, she saw images of her mom being dissected. Today, publishing a patient's medical records without permission could break federal law. But back in the 1980s, there were no such laws. The U.S. federal government enacted the Health Insurance Portability and Accountability Act, HIPAA, in 1996. This made it illegal for doctors to publicly share patients' names and records without consent. The second controversy around HeLa cells is about the right to informed consent. Should doctors get a patient's agreement beforehand when extracting cells from their body? Today, in cases like Henrietta's, doctors must get consent. But back then, things weren't so strict. In February 1951, while Henrietta was unconscious on the operating table before surgery, doctors took 2 tissue samples from her cervix without telling her or her family or asking if she wanted to donate cells. They just did it. First, in 1951, this wasn't illegal or against ethical guidelines. Back then, informed consent wasn't really discussed in medical circles. Plus, Hopkins Hospital provided free treatment to poor Black patients like Henrietta. Doctors in that era often experimented on charity ward patients without full disclosure, reasoning that since they got free care, using their tissues was fair payment. But existing laws did require consent for post-mortem procedures like autopsies or sampling. So after Henrietta died, doctors asked her family's permission for an autopsy. Though reluctant, her husband agreed after being told tests might help his children, while the real reason was Gay's lab wanting more tissue to see if it was immortal like HeLa cells. The hospital likely never explained this truth about the non-consensual experiment. Even more extreme cases happen in the U.S. In 1956, virologist Chester Southam injected HeLa cells into over 600 prisoners and patients to test if they caused cancer, just calling it cancer tests. He simply told subjects it was cancer testing without full disclosure. Many developed tumors at injection sites afterward. Southam only got a suspended license, regaining his reputation within a year with no real consequences. Compared to then, the US now has robust professional regulations. Today, doctors must get consent for tissue collection specifically for research. But during diagnostic procedures like biopsies, consent isn't required for incidental sampling. Today, when most Americans get a blood test or have an appendix removed, that tissue doesn't necessarily go straight to the trash. It might be preserved by the lab. In such cases, the removed tissue is classified as medical waste, eliminating the need for patient consent. Now let's examine the third controversy sparked by HeLa cells: ownership of body tissues. Are human tissues a patient's personal property? This question sounds simple at first. Of course, my cells belong to me. But what you believe and what the law recognizes are two different things. To date, no U.S. court case has clearly established whether individuals own their tissues. While tissue is inside your body, it's undeniably yours. But once removed, it's unclear. A 1980s case is typical. A cancer patient named Moore had his cells used to develop a new cell line, leading to crucial proteins and a patent. When getting his consent, the doctor claimed it wasn't for commercial purposes. Moore believed this deception equaled cell theft, essentially stealing money. He sued, becoming the first American to legally challenge body tissue ownership. But courts ruled in the doctor's favor. Once removed, tissues become medical waste that anyone can acquire and sell. So how should we approach this dilemma? Upon reflection, it's complex. Scientists take minuscule tissue fragments, not entire limbs or organs. Yet, people feel even that tiny fragment is theirs? How can it be taken so freely? Opinions remain divided. Ethicists, lawyers, doctors, and patients supporting ownership hope lawsuits will establish new laws. They await a day when people control their tissues like their money, by personal choice. Opponents argue that giving patients control over their tissues is short-sighted because public responsibility toward science matters too. People have an obligation to contribute tiny resources from their bodies for scientific progress, since everyone benefits eventually. Plus, if individuals owned tissue rights, scientists would face way more red tape getting cells, potentially hindering research. So tissue ownership remains unsettled. But this leads to the fourth controversy. Regardless of ownership, if commercial profits come from studying someone's tissue, shouldn't the contributor get compensation? This question also lacks clear answers. Just looking at HeLa cells, their commercial impact is massive. Initially, scientists just bought cells for research. Hopkins and Gay never profited. For example, in 1952, the Polio Foundation built a cell factory producing 20,000 tubes weekly for 23 U.S. research centers. This factory supplied labs for experiments only, no commercial sales. But when they realized HeLa cells grew so fast they'd never run out, they expanded operations. Scientists could then order HeLa cells for just $10 plus shipping. Soon, the factory couldn't keep up with demand. Years later, for-profit cell distributors emerged, followed by biotech companies. Since these companies sold multiple biological products, HeLa cells' exact profit contribution remains unquantifiable. We only know current prices range from $100 to $10,000 per tube. Estimates suggest HeLa cells helped create a multi-billion-dollar biological materials industry. Yet Henrietta's family never saw any benefits. They still live in poverty and poor health. Henrietta's family all have high blood pressure and diabetes, often unable to afford medical treatment. Her daughter Deborah's situation was the worst. She was constantly troubled by the HeLa cell issue, worrying someone might clone her mother, while also getting bombarded with calls and questions from strangers. In the end, it was all about the HeLa cells. She was diagnosed with anxiety, depression, and other illnesses. Every day, she had to take 14 different medications. Deborah said many people thought her family got rich from the HeLa cells, believing they must be millionaires. But in reality, they never saw a penny. When Deborah died in 2009, her bank account had no savings at all. Despite numerous precedents and cases, the HeLa cell controversy has also drawn a lot of media attention. But the Lacks family never took it to court. Henrietta's son said they didn't want to cause trouble for scientific research. Although they didn't understand how it was done, they were still happy about what their mother did for science as she helped many people. However, they also hoped that as family members, they could get some benefits. Should Henrietta, as the cell provider, or her family receive financial compensation? Currently, this is still a legal gray area. There are no clear regulations when it comes to financial compensation. Both opponents and proponents share the same concerns. If money is directly compensated to individuals, someday there will be people who exploit human tissues for profit. For example, someone might demand sky-high prices for their tissues, which could hinder normal research. Actually, the issue isn't whether human tissue research should be commercialized. It's already a fact and will continue. Without the commercialization of human tissue research, the drugs and diagnostic methods we rely on couldn't be developed. The key is how we should handle this commercialization. Scientists should tell people that their tissues might be used for profit, inform them in advance about the uses and potential commercial benefits of the cells they provide, and obtain consent. This could reduce post-event problems. All right, we've now covered the 4 controversies raised by HeLa cells: privacy protection for cell providers, the right to informed consent, tissue ownership, and whether individuals can benefit financially from commercial development. Most of these issues remain unresolved debates, consistently hot topics in academia. All right, we've covered the story of HeLa cells. I should note this book was published in 2010. Therefore, the current situations mentioned refer to pre-2010 conditions in the US. Though long gone, the publication of The Immortal Life of Henrietta Lacks allowed this voiceless woman to fully share her story for the first time. We see science isn't just cold experiments and data. It's backed by human contributions. In recent years, academia has realized research shouldn't be the sole focus. Donors like Henrietta deserve respect and protection. Some med school professors honor Henrietta at annual HeLa conferences, and one even bought her a tombstone. Social activists plan to establish a Henrietta Lacks museum. Finally, I'll conclude today's discussion with a passage from the book's epigraph. This quote comes from Nazi doctors and the Nuremberg Code. We must never see any person as an abstract entity. Instead, each individual is a complete universe with their own secrets, treasures, and unique pains and triumphs. That concludes our discussion of The Immortal Life of Henrietta Lacks. Reading notes are below the audio. See you tomorrow!